Muscle contraction is based on four functional characteristics associated with muscles: excitability, contractility, extensibility, and elasticity. Pay attention to the differences in definitions between extensibility and elasticity, because it's not what we typically think of when we think about a rubber band that's elastic, etc. Again, this property here is what's tied to the action potential, the electrical gradient that is responsible for contraction in the first place. Last but not least, contractility here. This is what we talk about when we talk about muscles shortening, literally flexing and extension, and all of those movements result in muscles literally becoming shorter than they were, and that shortening is what provides the force, utilizing the lever action of the bones to produce movement. Okay, some quick anatomy review here in terms of blood and nerve supply. As was established with the electrochemical gradient and the presence of the sodium-potassium pump, it is a huge source of energy consumption. That means you have to have ready access to the energy, meaning we have to have access to blood, and then we have to have access to the nerves, because the nerves are what release the acetylcholine that causes the depolarization that leads to contraction in the first place. So notice how blood and nerves gain access to the muscles and the individual muscle fibers. And all of that has to do with how they break down and they branch within the muscle. Now remember that a muscle, as we know it, the biceps brachii, the latissimus dorsi, the rectus femoris, each muscle is an individual organ within the muscular system. So that means each organ, to some extent, is independent of everything else. It stands alone. It's isolated by the fascia, by the paramycium, the epimycium, and the endomycium. And the endomycium and the paramycium play a role within the muscle by kind of providing reinforcement and allowing that shortening of the muscle or pulling the muscle to reinforce the actual attachment by the epimycium to the periosteum of the bone. So this is when you pull on rubber, or we talked about it when we did the lab, and you pulled on the string and the entire arm had to move or the entire leg had to move. The epimycium is what's responsible for that. The paramycium, the endomycium provide that framework within to kind of help hold the whole thing together. So we know energy is required, we know innervation is necessary, because that's what sets up this whole change, the release of the acetylcholine across the synapse, and this combination where we see the nerve come in to the individual muscle cell, the muscle fiber. Remember, talking the fiber here, not the myofilaments, is what we term the neuromuscular junction. Now, as a reminder, our junction are the axonal endings from the neurons. They come into contact, well, not physical contact, but we create that synapse with the sarcolemma. This is what's known as the motor end plate. So we have a few more terms that you have to be familiar with. One of the most important in terms of contraction is this idea of a motor unit. That's going to play a very big role as we discuss through this particular section and we talk about recruitment. For now, a motor unit is what's responsible for the number of cells that will be contracted by a single neuron stimulating parts of a muscle. In other words, our muscles are not all or one, and you kind of know this because if you put a pencil in your hand while you're watching and you lift up and you bend your arm and you flex your bicep, you don't use a lot for that movement. But if you put your anatomy book, a little more is required. If you actually put a 15-pound weight in your hand, that's more than your anatomy book. Now even more force is required to overcome the weight in your hand. And so what we see is we see motor units, more and more motor units being stimulated. Now, you can also think of a motor unit in terms of fine versus coarse movements, or perhaps massive movements. In other words, Riding is a fine motor skill as opposed to walking, which is a massive movement. But I'm getting a little ahead of myself. But keep that in the back of your mind. So this is why motor units vary in size from just a few muscle fibers to thousands or hundreds. And again, it has to do with what kind of movement are we producing, what kind of skill is required. So when we talk about our cells being stimulated within our hand, for example, as opposed to our thighs, there's a huge difference in the skill required. Walking's not a fine motor skill, but riding is. Now, notice the other thing that's really important that we don't think about, because your initial thought, or at least my initial thought, was, well, they're going to be all grouped together, and they're not. When we talk about a motor unit, it's literally spread throughout the muscle. The importance of this makes sense as soon as you think about it. If you don't have a motor unit spread throughout the muscle, and let's say if you talk about your bicep and just like the top quarter inch is stimulated because you're just lifting a pencil, you're not going to get any movement. It's like the muscle would be working against itself. You'd have tearing. So by having the motor unit spread throughout the muscle, when the motor unit is stimulated, the entire muscle will contract, even though not every single muscle fiber within it is stimulated to contract. All right? Now, the stages of contraction are threefold. You have to have the action potential, you have to have the contraction, and then you have to have the return to the resting state. Now, what's kind of cool about this is what we know about depolarization and repolarization already. The thing to remember now is depolarization and repolarization is no longer just the sarcolemma. I got that a little crowded. We're talking also about what's happening internally at each myofibril and the role the sarcoplasmic reticulum plays. So look back at your diagram where you see how the sarcoplasmic reticulum surrounds all of our myofibrils, and we have these T-tubules. What you have to remember is because of the T-tubules, because of the sarcoplasmic reticulum, the SR, the sarcoplasmic reticulum, is literally one with the sarcolemma. It's physically attached. So because it's physically attached, when we see the action potential spreading along the cell membrane, the sarcolemma, it causes changes to the sarcoplasmic reticulum. In other words, just like we caused a permeability change in the sarcolemma, you have an interior permeability change within the sarcoplasmic reticulum membrane. And that change allows calcium to escape. So remember that the role of the sarcoplasmic reticulum was to sequester calcium. In other words, the reason your bones store calcium is because we need calcium for muscles. So when our body, one of the most important places that we use calcium is within the muscle cells, because it's tied to muscle contraction. Okay, so segue back. So that means when I change the permeability of the sarcolemma, I have a subsequent permeability change within the sarcoplasmic reticulum, and suddenly I have calcium released. This is important because remember that we have troponin covering the actin binding sites. This is where the myosin would bind to the actin. This is back. Review your microscopic anatomy real quick if you need to. So pause this and go do that and then come back, or simply review your notes as we go. The thing to recognize and the thing to remember, when anything binds to a protein, it changes its shape. That's huge. So when the calcium binds to the troponin, it causes the troponin and the tropomyosin, therefore, to change shape. And the second thing is anytime a protein changes shape, its function changes. So by changing the shape, we change the function. And changing the function allows these binding sites to be available to the myosin, and now the myosin can bind to it. Now, this is where we see the second high energy use of muscles, because this cocking of the myosin head, in other words, you can think of it as taking your hand, if you put your hand on your ears and then you push it all the way so that your elbows are to your ears, That's like the cocking of the myosin heads. So as soon as you do that, and then I fly forward, if you imagine holding on to pipe or something from behind you and bringing it forward, that's what happens with the myosin and the actin. So you get this pulling, this sliding of the filaments, and this is the source of the sliding filament theory, the idea that the actin and myosin slide past each other. Now remember that the actin and the myosin, I'm going to jump ahead a little bit here, when they slide past, okay, it binds to it, ATP cocks it, it binds to it, it slides past. Remember that the actin is what's attached to the Z lines. So when the actin is pulled to the center of the sarcomere, the whole sarcomere shortens. If the sarcomere shortens, that means the entire muscle shortens. So it's sort of like I have a sarcomere attached to sarcomere. It's like when you pull apart one of the, oh shoot, what are they called? Slinkies. When you pull apart a slinky and then you let it go, it all collapses together at the same time. That's what happens with the shortening of sarcomeres within a muscle, is I have all of these actins sliding past each other and back towards each other, and that's what produces the ultimate shortening, the contraction. So again, when the myosin pivots past, so in other words, you take your hand at your elbows or at your ears and you suddenly pull your whole arm forward, that's what's going on. That's the sliding of the actin past the myosin. We get the shortening of the sarcomere. This is what's known as the sliding filament theory. Now, the thing to also recognize is how many times have you ever exercised to the point that suddenly you get cramps? Okay, part of that's going to be due to the buildup of lactic acid, which we'll talk about in the next section. But the other reason is if you start running out of ATP, the cross bridges won't release. Now remember that the cross bridge is the attachment formed when the calcium binds to the tropin and tropomyosin, it moves it out of the way, the myosin binds to the actin, that's the cross bridge, all right? So this whole attachment that occurs right here is the cross bridge, and I have to have ATP to break the cross bridge. So that means every time, and now think about this in terms of the context, in terms of small versus large muscles even, every time a single myosin head binds to a single active site, 1 ATP was required to cock it, 1 ATP is required to detach it for every single myosin head. Now go back and look at that cross section of the muscle. So again, see how we have each muscle kind of this category, this pull down from the whole muscle, this small part. And so we have the single muscle fiber, just the single cell. And so now, and then with each of those, we have all of this circle of every myosin surrounded by actin, every actin surrounded by myosin. And again, every head has to have ATP. So again, huge ATP requirements. Now, continuing with contraction, trying not to get too many segues in here, we have then the ACH has to get broken down. That's the acetylcholinesterase that's going to break down the acetylcholine that caused the initial permeability change at the sarcolemma. So in essence, again, we have these 4 basic steps. So you have to have, these are in essence the anatomical changes behind the shortening of muscle. So you have a series of steps. Pause it and copy these down. So this is our start and this is our end. So again, we have to be able to start contraction, do contraction, and then end contraction. Just like what was observed with the action potential, contraction is all or nothing. Now, this is a great image that puts it all together in terms of the physiological changes associated. Again, you can see how the T-tubules are what make the sarcoplasmic reticulum one with the sarcolemma. So when I have a change in the permeability of the sarcolemma, that causes a change in the permeability of the sarcoplasmic reticulum. I see the diffusion of calcium. So again, by creating gradients, diffusion is easier and it leads to massive changes inside the cell. We then have the resequestration of the calcium back within this, and this is what will lead to the end of muscle contraction.